Hey, good morning. It's my pleasure to introduce Jackie Smith or Jacqueline Smith. Um, Jackie got her bachelor's. She graduated cum laude from University of North Carolina at Wilmington in environmental science with a chemistry minor, where she came more from a biology background. She's now working on her MS degree in ecosystem health for her MS. She's worked She's been at, she worked as a biological research technician at Beltsville for a couple of years. And in the last couple of years, she's been working on her MS degree. And so she's part of a large project looking at farm ponds, where she's doing the phytoplankton part of it. Someone else is doing the E. coli part of it, and someone else is doing the drone's hyperspectral photography part of it. And these will all merge together in the end. So Jackie is co-advised by Dr. Yakov Pachevsky and myself, and she's doing well. She's originally from New Jersey, but you can't tell it generally. She's very nice. So I present Jackie Smith to you.
Thank you for the introduction. Hello, everyone, and thank you for joining me today. My talk today is going to be on my master's work, and the title is Dynamics of Phytoplankton Populations within Irrigation Ponds. I, of course, did not do all this by myself. So I do have my head advisor, Dr. Hill, co-advisor, Dr. Pachevsky, as well as Dr. Kangas and Jennifer Walney, who is now at the FDA. So kind of give an outline of what I'm going to be talking about today. I am going to go over some background and importance of the freshwater phytoplankton, as well as quite a bit of methodology. And then my thesis is technically 3 chapters. So the first chapter is on spatial and temporal variability of these phytoplankton groups within the irrigation ponds. And then also chapter 2, I have the environmental controls and predictors of these phytoplankton groups within the ponds. I also have a 3rd chapter, which is still kind of in progress, looking at the coexistence of cyanobacteria as well as E. coli in managed irrigation ponds. So this is still in progress, and today's talk, I'm only going to be diving into chapters 1 and 2. I will not be talking about chapter 3. So a little bit of background, phytoplankton, they are free-floating microscopic algae and bacteria as well, which are found in most water bodies. And this includes fresh, marine, as well as brackish. I just recently learned even sea ice technically. So they are primary producers and they're essential to most aquatic food webs. They're highly diverse, very sensitive to changes within their surroundings. So some species of phytoplankton are even capable of producing poisons called cyanotoxins. There's a couple other toxins as well. And some of them are actually some of the most powerful natural toxins known to man. And furthermore, there's no known antidotes for these. And it's basically just symptom-based or palliative care. Some species are even known to form large blooms, aquatic blooms, and these can actually lead to hypoxic conditions with ecosystem-wide implications. So why are these guys important? Basically, they're actually really excellent bioindicators. So due to their highly sensitive, very diverse nature, they can not only be an indicator of just a basic trophic level or trophic state of a water body, but they can even indicate water quality changes and just the overall health of a water body. As well, there is kind of a human health aspect to this. So cyanotoxins can be transferred from irrigation waters and transferred to the crops and even soils during irrigation. And there have been quite a few studies that have even showed that these toxins can be taken up by the roots of the plants and remain within the actual flesh of the plants, as well as remain in soils for extended periods of time. So the simple presence or absence of a species can even be an indicator of a water quality, of general water quality. So for example, presence of euglenoids, which are actually a part of the flagellate group, often associated and one of the first species to pop up after water body has gone through some form of degradation. As well, community composition has been used as a water quality bioindicator in the past. Plenty of studies have been done within lakes, rivers, reservoirs, even gulfs. However, there have been very limited work has been done with phytoplankton populations in irrigation ponds. So diving into the methodology here, the easiest way to kind of like break this study down is into two separate portions. The first portion being field monitoring of these irrigation ponds. So this consists of field sampling, lab analysis, as well as microscopy work and some data analysis. And then there's a second portion which consists of cyanobacteria culturing, as well as some small microcosms. That is more related with chapter 3. So we won't be going over any of the cyanobacteria culturing or microcosms. Chapter 1 and 2 fit very well into the first portion of this study. To go over some basic field sampling. So we sampled 2 working irrigation ponds in Maryland, one of them in Germantown, Maryland, as well as Y Island, Maryland. Both of these were right around, I think they're both just over an acre. They seem drastically different, but they're about the same size, just different shapes. And we performed dense grid-like sampling at both of these ponds. Both nearshore and interior water samples were taken, and interior was taken on a little boat displayed there, as well. A hand grabber was used to get the nearshore samples. And sampling locations remain the same throughout the entire study. We used a GPS with about 2-foot accuracy, although most of the time it was a lot better than 2 foot. That was just what the company guaranteed, just to ensure that these locations stayed the same through the study. So in the field, lots of measurements were taken. Majority of them came from our YSI XO2 water quality sonde. So this collects temperature, specific conductance, pH, dissolved oxygen, phycocyanin, turbidity, fluorescent dissolved organic matter, as well as chlorophyll measurements. And we also did some other measurements out in the field. Those were just water depth, as well as Secchi disk measurements. And we have a photosynthetic active radiation meter and also a submerged light meter, which we took at 3 depths. One at the surface, one at about 7.5 centimeters, and the next one at 15 centimeters. So some lab measurements were performed after field sampling and in the lab. So samples were run on a fluorimeter for color dissolved organic matter, phycocyanin, as well as in vivo or whole cell chlorophyll. And we also performed extracted chlorophyll analysis on these samples using the EPA method 445. So this was typically all done within the same day. The only exception to this is the extracted chlorophyll analysis, which needed to steep overnight in acetone. But for the most part, all of our numbers, we got that same day. A small subset of water was taken off and portioned for microscopy analysis. We preserved these samples with a Lugol's iodine solution to get it to about a 1% iodine concentration in the final volume of water. And then at that point, they were just stored in the dark in a fridge at 4 degrees Celsius until we were able to process them on the microscope. This varied, so usually somewhere between like the 3 to 8 month mark was about when we were able to process them. So all the microscope samples were analyzed for 5 major phytoplankton groups. Those groups are diatoms, dinoflagellates, chlorophytes, flagellates, as well as cyanobacteria. We further classified diatoms into 2 orders, centric and pennate, and then we even further classified cyanobacteria into 3 most common orders for freshwater, which were Nosticales, Croococales, and Oscillatorialis. And if there was a bloom or any type of specific species that I kept seeing pop up in the microscope, I would try my hardest to go down and identify that to species level and just leave it within the notes. Usually this was done for cyanobacteria because out of utter interest, I wanted to see if they were a toxin or non-toxic version of cyanobacteria. So a lot of people, you can mention algae, they probably know what it is, but when you break them down into these smaller groups, some people might not be as familiar. So I'm going to do a very brief overview of these 5 major groups. The first one being diatoms. So these guys, they have a cell wall made of silica. They are considered true algae, so they are eukaryotic. And they have 2 distinct shapes, that being radially symmetric and bilaterally symmetric. And this correlates with the 2 orders of the centric and the pennate. They typically are single cell, but I often find them in colonial versions as well. And there are a few species that are toxic. Some of them are associated with shellfish poisoning, so that would be with the domoic acid. The next group here is dinoflagellates. So these guys are true algae as well. They are eukaryotic. They typically have what I like to call an armor surrounding their cell. It's not the same as a diatom, but it's nice and structured enough so that when we preserve them, they actually preserve very well. Almost completely single-celled. They have 2 flagellates as well. And toxic species do exist. This is what we commonly refer to as the red tide. So the next group here is your flagellates. These are true algae as well. They are single-celled or colonial. And these guys are the hardest to identify when they're preserved because most of them do not have any type of structured cell wall. Majority of them are what we call a naked cell wall, and they do not preserve well. So they kind of, after they've been preserved, they kind of look like a ball of mush in the microscope. But the other ones, like I have a couple pictured here, they can be scaled, like in the upper right-hand corner. They can also have calcium carbonate, like a cell wall, and as well as silica. So the chlorophytes, this is probably the most iconic group. So this is when you say algae, this is what people think of. They are eukaryotic, they're true algae. They can be either single-celled or colonial. And these species are even capable of forming blooms, similar to the ones I'm going to talk about next, which are cyanobacteria. So these guys are actually not true algae. They are prokaryotic and they are considered bacteria. However, they are photosynthetic. And oftentimes you'll hear them being referred to as blue-green algae, which scientists these days are trying to stop the use of that term because they are not technically algae. And they usually are colonial in the environment. So when you are taking like a water sample from a nearby stream or water body, a lot of times they will show up in clumps or colonies. But when you culture these guys, sometimes they can tend to be single-celled. And they are bloom forming. And as well, they produce quite a few different types of toxins. You have your microcystin, your saxitoxin, anatoxin, and then cylindrospermopsin are some of the most common ones. So as you can imagine, that's a lot of data. So for analysis purposes, we did some grouping. We grouped together the diatoms, so the centric and the pennate diatoms. And then we also decided to group together chlorophytes and flagellate as green algae. However, it's important to note here that this is a rather large assumption that we are making because not all flagellates are actually considered green algae. Probably only about half of them are. Some of them are considered, but the other half are not. So there is an assumption here when we decided to group them as green algae. And then together, all 3 of the orders of the cyanobacteria we decided to group. So for the actual quantification, we used a modified Utermal method. And this is a pretty common practice in this area. So this method is also used by Maryland Department of Natural Resources and even Virginia Department of Health for their freshwater systems. And how this typically works is you take 2 to 3 milliliters of preserved sample, and that would sit in a chambered covered glass slide for about 30 to 45 minutes prior to counting so that the cells can settle to the actual glass on the bottom. And then a Nikon inverted microscope was used with the 40 times magnification for all the samples in this study. So it's kind of hard to describe how I actually go through and count these on the microscope. So I made this little diagram to try and make it as easy as possible to understand. So the large peach or orange colored square is going to be the actual chambered cover glass slide. And the blue squares are going to be my fields of view as if I was looking through the microscope. The arrows are going to show you the movements that I actually make through the slide. And every time I finish counting a field, to move to the next field, a movement of down and to the right or to the left, whatever I'm deciding that day, always has to be made. So it always has to be down and to the side. And this helps to prevent any type of overlap of the fields of view. And that way it limits any like double counting or counting an organism more than once. And then this pattern continues through until I either reach 200 cells or 20 total frames. And then at that point, that sample has been completed. So this provided us with quite a lot of data. We have 2 years of sampling. In 2017, we had 11 sampling dates. In 2018, we had 12 sampling dates. And these were all done during the growing season as well. Pond 1 has 2 ponds. So Pond 1 has 23 sampling locations, and then Pond 2 has 34 sampling locations. This comes out to be well over 700 samples. And for every single one of those 700 samples, we have biological and physiological parameters for each location. So in 2017, we collected 9 parameters for each location, or excuse me, 19 parameters for each location. And then 2018, 26 parameters. And then in addition to that, all of those 700 samples got assessed on the microscope and looked at for those 8 phytoplankton groups. My eyes were not happy with me, but I got them done. So moving into interpreting all the data, I'm first going to discuss chapter 1 and the spatial and temporal patterns that were found. So the major objective here was to determine if there were any patterns in the total populations of these phytoplankton groups throughout the pond on a daily basis, as well as seasonal basis and even multi-year basis. So why does this matter? So phytoplankton are an essential part of the habitat in which pathogenic and indicator microorganisms live. So if we can establish a relationship between the phytoplankton and the E. coli, we can possibly use these sensing technologies such as like hyperspectral imaging or even in situ sensing to help design monitoring techniques and protocols for E. coli as well as pathogens. As well, phytoplankton can be a source of these cyanotoxins and other harmful substances that can potentially be transferred to crops and soils. And knowing the distributions of the phytoplankton can aid in developing these monitoring strategies for these ponds. So the goals of the first chapter were to try and establish these patterns within the community of phytoplankton. So were there differences between the nearshore and the interior samples? How did these populations behave over multiple sampling years? What about within a single sampling season? And are there stable spatial trends across the pond, or do these zones or hotspots exist? As well, what happens with the community composition proportions through an entire sampling season? We decided to use mean relative differences, or MRDs, to assess the spatial and temporal stability. This would allow us to understand how these groups behave through the sampling season and even over multiple sampling seasons. As well, it indicates any types of hotspots or zones of consistently higher or lower phytoplankton concentrations. And we also used Pearson correlations to help utilize or to compare the spatial patterns between years, as well as compare the spatial patterns of water quality with the spatial patterns of phytoplankton groups. And then all computations for this chapter were run in R for the most part. So why do we choose mean relative differences? The one main reason here is they are one of the most common methods for analyzing for temporal and spatial trends. So the easiest way to put this, a relative difference is how a singular location differs from the pond average, and then that's calculated by taking a location concentration, deducting the pond's average concentration, and then dividing it again by the pond's average concentration. So then you have your relative differences. We have a relative difference for each location, for each sampling date. They end up getting averaged at the end of the summer, and that gives us a mean relative difference for the entire sampling season. And this shows locations in which the concentration are consistently higher or consistently lower than the average across the pond. So this produced quite a bit of data. So there was a lot of graphs. I want to say there was probably close to 14 graphs that came out of the study. So for presentation purposes, I am only going to be showing you one. Here I have an example of Pond 1 diatoms, their mean relative differences. And this is actually for combined years. So this is for 2017 as well as 2018. Looking at this, you see there's green, yellow, and red. The green locations indicate locations with significantly lower than the pond average, lower concentrations. The higher locations with the red symbols indicate areas with consistently higher than the pond average. And then the right in the middle there with the yellow symbols, that indicates that those locations were approximately the pond's average. So an MRD value of 0 is basically just stating that that location is about the pond's average. The temporal stability was also assessed by using the size of the error bars. So the larger the error bar for a location, this means that that location is actually less stable. And the smaller that error bar is, indicates that that location is more stable across a season or across years. So all of this was done for 2017, 2018, and then we also combined both years and did a 2017-2018 data set. And we were able to determine that these major phytoplankton groups are, for the most part, temporally stable for both years individually as well as combined years. And we were also able to distinguish some zones for cyanobacteria. And there was a very prevalent pattern of these differences between nearshore waters, nearshore locations, and interior sampling locations. So to dive into some of these results and give you kind of a bird's eye view of the ponds and a better way to kind of visualize this, I do have a picture here of Pond 1. I have the locations that are the highest and the lowest in regards to mean relative differences. So the low MRDs are associated with the green squares, and then the high MRDs are located in the red squares. So for Pond 1, almost all of the low MRDs were located within the interior waters, and high MRDs were mostly nearshore locations and were associated with some type of flow change or flow conditions. For example, if you look in the southern portion of the pond here, we have location number 1, 10, and 8. So these are all located for the most part slightly downhill and right adjacent to a crop field. So they likely receive some type of runoff during large rainfall events. As well, location number 5, up here, there is a inflow pipe, which pulls water from a nearby pond, which is creek fed. And location number 23 is actually a really shallow location. It has lots of aquatic vegetation, and it also has a really small inflow point, which is fed by an ephemeral creek. Now looking at the diatoms, for the most part, same thing here. All the low ones seem to be within the interior of the pond, and the high ones were all nearshore locations. And again, they were all, for the most part, associated with some type of flow change or flow conditions. So we have location 1, which is right adjacent to the crops, 5 with the inflow pipe, and 23 with the inflow from the ephemeral creek. And then spot number 6 is showed up this time for a high MRD. Spot number 6 is actually the location of the irrigation pump for this farm. And if you can see in between, it's kind of hard, but in between 6 and 12 locations, there's a small white dot, and that's actually the intake for the irrigation pump. So the cyanobacterial spatial patterns were very interesting. We were able to discern a zone here. This is pond 2, and it looks like in the northern portion of the pond, there seems to be a hotspot of cyanobacteria. The only one that does not agree with this is location 21 towards the southern portion of the pond. However, this location is actually pretty different from the other ones surrounding it in that it's very shallow, and this location as well has quite a bit of aquatic vegetation. But in regards to the zone in the northern portion of the pond, it's rather interesting because location 12 actually has a culvert there. And anytime anytime this pond gets a decent amount of rain, there's usually some type of inflow from a nearby wooded area and an ephemeral creek. So we also wanted to examine interannual mean relative differences, so between year patterns. To do this, we used Pearson correlations. And for the most part, green algae as well as cyanobacteria didn't really show any type of interannual similarity. However, diatoms did show a little bit of a similarity from year to year. Both ponds had an R squared of about 0.3, I believe it was. And while you can see they don't necessarily match up perfectly, there is a nice trend going on there. So for diatoms, both ponds, there seemed to be a trend between years, but that was not seen with the green algae and cyanobacteria. So the last portion of this chapter, we decided to try and compare the water quality patterns with the phytoplankton patterns to see if we could make any correlations. We used Pearson again to assess the water quality MRDs and the phytoplankton MRDs. And what we found is certain groups correlated pretty well with water quality parameters. We chose an R squared of about 0.4 or greater in order to list them as a moderate correlation. And in this table here, anything highlighted in green is considered a moderate correlation. The biggest takeaway is while there was quite a bit of correlation between the water quality parameters and the groups, they seem to be different between the ponds. So for example, pond 1, green algae, almost all of the water quality parameters it correlated reasonably well with. And then for pond 2, that was kind of the opposite. Green algae didn't correlate very well at pond 2, but diatoms and cyanobacteria did. So these findings can indicate a few things. One being that, so we do have these relationships between the water quality and these different algal groups or phytoplankton groups. It seems like it might be largely site specific though, and shouldn't be assumed to be the same for different water bodies. As well, this is good news in regards to trying to assess for phytoplankton. It is a lot easier to get a pattern for water quality than it would be to get a pattern for phytoplankton, where that method is a lot more intensive, requires a lot more time, specifically trained personnel, than a water quality sonde. So some major conclusions for chapter 1. The biggest takeaway is the 2 sampled ponds that we did, they didn't exhibit any type of spatially homogeneous phytoplankton populations. So they were different. These zones existed for different sampling locations, which were either consistently lower or consistently higher than the actual pond's average. And as well, these spatial patterns of water quality seem to correlate pretty well with the spatial patterns of the phytoplankton groups, although there is this dissimilarity between the 2 ponds. So what are the major implications for this? Sampling locations and your irrigation intake placement shouldn't be just an arbitrary decision. So these phytoplankton populations can be drastically different depending on the location within the pond. And this also means that less intensive monitoring methods like in situ sensing might be able to be used rather than this more demanding microscopy work. And this can be efficient for monitoring, prediction, as well as the management of these ponds. So then moving on to the second chapter of my thesis, it's attempting to find controls as well as predictors for these phytoplankton groups. And to do this, I enlisted the help of machine learning methodology. So the major objectives of this chapter were to determine if there were any influential water quality variables that were associated with certain concentrations of major phytoplankton groups, as well as to try and develop a machine learning model to help predict the density of these phytoplankton groups using these water quality variables as inputs. So why are we doing this? What's the importance behind this? So a predictive relationship, it needs to be built when there's a couple situations. So either the number of your potential predictors is too high or you have a lot of them, or when you don't necessarily know which one of them are going to be important. And especially when your relationships are not necessarily linear relationships. So this is a really good situation for when machine learning might be the best option rather than using these standard statistical methods. Random forests were specifically chosen due to their simplicity and ability to produce accurate results with minimal tuning. They're also one of the most commonly used algorithms for classification and regression tasks. So the first here is going to be my regression tree analysis. So regression tree algorithms, they help to identify the most influential water quality parameters and as well help determine that water quality parameter's ability to find the most accurate tree. This does this by splitting the data into two groups, the first being data that are as similar as possible, and then the second group being data that are as dissimilar as possible. And all the regression trees were computed in R using the rpart package. I do have an example here on the right, and this is of diatoms in pond 2 for 2017, only using physical parameters. And how the tree works is at the top of the tree, you can see 100% of the observations. The average for those diatoms was 6.3, a log of 6.3 cells per liter. And the first determining factor, or the first influential parameter, is going to be your pH. And if the pH is greater than or equal to 8.8, you would follow the left line, and that would drop you into the first bin there where it's about 32% of the total observations. And the average of the diatoms would be about 5.7 log cells per liter. Now, if the opposite case is true, if the pH is less than 8.8, then it would drop you into the bin to the right. And these leading predictors up at the top are the most influential water quality parameters for this group. So the trees themselves are not necessarily good predictors. They do identify what these influential parameters are, but their prediction capabilities by themselves are not necessarily very good. So that's why I enlisted the help of random forests. The random forests make regression trees by randomly splitting the data into two data sets, a training data set and a validation data set. And it runs many, many trees in order to form a random forest. So it takes the average of all those trees in order to form a prediction. And there are some precautions built in so that it doesn't overfit. So that is nice to have in there as well. And so all of this was computed with R, and it was the random forest package that was used for this. So a little bit of, there's a couple of abbreviations before I move forward, just so you know what I'm talking about in regards to the results for the regression trees and the random forests. We used two input data sets. So the first one was the physical parameters. So if you see the letter P, that stands for physical parameters. And that is basically your temperature, dissolved oxygen, turbidity, pH. And then we also used PB, which is physical and biological parameters as an input. And that is basically the physical that I was just talking about. And then we also added in the biological parameters, which is basically our chlorophyll measurements, in addition to color dissolved organic matter and fluorescent dissolved organic matter. So the results from the random forests are pages and pages of tables. And instead of overwhelming you with them, I decided to summarize the results. And if anybody has like questions or they wanted to see tables or any error metrics or important variables, you can always contact me, or it can be read in my thesis when my thesis gets published. So the quality of random forest predictions, they depend on a few things. Firstly, the duration of the observation period, as well as the relevance of those inputs and which ecosystem, which in our case would be which pond, as well as the size of the data set. So our accuracy, it seemed to follow a trend, or a pattern, should I say, and green algae tended to have the best accuracy, followed by diatoms with a little bit less accuracy, and then cyanobacteria actually had the worst accuracy out of the three. And between ponds, the random forests seemed to produce less error at pond 1 and a little bit more error at pond 2. And the addition of these biological parameters, it seemed to increase the accuracy for the most part across all the groups and both ponds. And similarly, multiple years didn't necessarily increase the accuracy. So the accuracy of 2017 or 2018 wasn't necessarily increased when we decided to run them together. The major output of a random forest are these important variables. So for green algae, diatoms, and cyanobacteria, these important predictors were pretty similar between the ponds, the major ones being temperature, dissolved oxygen, conductance, as well as pH. And beyond that, similarity between the groups and the ponds for these physical parameters. So these 4 specific ones kept showing up as the top or most important predictors or variables between both ponds and all the groups that were run. There was a decrease in similarity between the ponds and the phytoplankton groups once those biological parameters were added in, however. But as well, there was pretty minimal change. Again, same thing with the accuracy. There was minimal change once we added in the multi-year data, and that was with the physical parameters only. So all of this was actually run as well with cyanobacterial abundance. So up to this point, everything has been shown in a concentration of phytoplankton, so cells per liter, and ours typically ranged from about 4.5 to 8.5 log cells per liter. The abundance is a little bit of a different way to go about looking into this, and we took the total cyanobacteria and the proportion of that compared to all the phytoplankton in the pond. So these values ranged between 0 and 1. And we ran the forests and the trees on them, and the results were pretty interesting in that cyanobacteria abundance had really good accuracy for both years as well as both ponds. And the important variables seem to be the same as well. No change there with ponds or within years, and they seem to be about the same as the 3 major groups. So some major conclusions from chapter 2. Random forests seem to provide some pretty accurate predictions for green algae in both ponds. As well, important predictors were rather similar, not exactly identical between ponds and groups, but they were pretty similar across all of the phytoplankton groups and both ponds. As well, minimal changes occurred in these variables and even the accuracy when we added in that multi-year. So a singular year wasn't necessarily better than having 2 years worth of data run. And cyanobacterial abundance had really good accuracy and similar predictors for both ponds and years. And I think the most important one here is these physical parameters seem to be the most important parameters for all of random forests. So what are the implications with this? So random forests might be a good option for prediction of green algae and cyanobacteria abundance from these water quality parameters. Most important variables within the random forest predictions tended to be these physical parameters, which means that we might be able to use these less intensive monitoring and prediction methods like in situ sensing or drone-based imagery. So the big takeaways from both of these chapters here is irrigation pond water is not a homogeneous mixture. For their size, how small they are, they do still experience these hotspots or zones or areas with lower or higher concentrations. And these spatial patterns of water quality parameters ended up correlating pretty well with the phytoplankton spatial patterns. And in situ sensing might be able to provide like a fast detection method for these phytoplankton populations. And as well, random forests performed well in predicting green algae as well as the cyanobacteria abundance. So I have some citations from my work here. And then just a couple acknowledgements. Obviously, my committee members, Dr. Hill and Dr. Pachepsky, and then also Dr. Kangas and Jennifer Wolney for teaching me almost everything I know phytoplankton related. And then as well, just USDA employees and interns, I have Matt Stocker as well as Billy Morgan, and then my two interns, New Lee and Nyla Washington. And then with that, are there any questions?
Really interesting talk, Jackie. Thanks so much.
Thanks.
So yeah. So please, I encourage everybody to put some questions in the chat if there's interesting things there. So I have a couple questions because, of course, you know, this is sort of not my area, but related enough that I was curious about things. So one of the questions I had, do you know how representative, like regionally, your phytoplankton characterization was to, other people in the area that maybe have done this on, like, for instance, larger water bodies?
So I have never looked at larger water bodies, but I was trained by someone who worked with the Chesapeake Bay. So she has taught me based off of what she was seeing in the Chesapeake Bay and like the species she would commonly see and other freshwater sources like around the Annapolis area. And so she provided me with like a PowerPoint of a lot of the species that she thought I was going to see. So they seem pretty representative, with the exception of maybe like the brackish water areas.
Gotcha. So like, so a subset of them, basically. I just wondered if these ponds are enriching for a particular, like there's a regional community and these ponds are kind of, enriching for a particular subset of the population, if you think that's a fair characterization.
I would say maybe. I don't know about like the actual abundance, but I feel like species wise, I feel like if I were to go maybe, I don't know, a couple miles down the road, there's a good chance I'm probably going to see the same species within that water body.
Yeah, that's kind of what I was curious about. Yeah, cool. So let's see. A question here. Excellent work. Excellent presentation. Says Adele, who will use the results of such a study and for what purpose?
So that's a good question. So this would probably come down to monitoring purposes. So someone like a farmer who is trying to assess the water quality of his pond prior to irrigating, he might be able to go down and, depending on what his pond layout is, and sample specific areas where he knows are typically hotspots, or to get a good representation of where his pond concentration is prior to irrigating, to maybe prevent like something like cyanotoxins or poor water quality being irrigated onto those crops.
So what are the rules about like that in terms of, the potential for irrigating with a toxin? I mean, what it currently has to be, what currently would someone have to do to ensure that that's not the case?
Usually that enlists sending samples out. So a lot of times this is done usually with an ELISA test, or I think it's gas chromatography. So these are not simple tests. So to see like actual toxin levels, this is you'd be sending samples out, they would be processing them, and then they would tell you a few days later what those toxins are.
Gotcha.
And if they're present, what levels they're at.
Yeah. So do you see it like the machine learning piece of this, that it could be, I mean, like eventually like a phone app, or would it still require like sending samples? Like what's your kind of what do you think the future of this is?
I think it might be like a surrogate measure. So you can measure if your levels of, say, everything checks out in regards to the physical parameters. Okay, you're good. But if they might come back as being like a high level, or there's a good possibility you might have a bloom of something, I think there might still be a need for like sending samples out at that point. But I think it would be kind of a preliminary check, and there is a first wave measure to double check.
Comment, Stephanie. This goes beyond Jackie's work, but that's part of someone's working on E. coli at all these same locations.
Right.
And then there's literally, there's also the hyperspectral that has hundreds of bands. So when this is all grouped together, it's going to be God knows how many samples.
Right.
And so the whole data set itself could be excellent for artificial intelligence and machine learning.
Right.
So it has a major goal that's beyond what can be done by any one graduate student.
Right. Yeah.
Okay, excuse me, I'm
Yeah.
sorry.
No, very interesting. Yeah. Yeah. Oh, let's see. So some other question. Do farmers typically do any kind of monitoring of their irrigation water before use?
Do you mean specifically in regards to algae, or?
I assume that's probably what Marty means.
So there's still like a lot of works. There are limits set up for like cyanotoxins for drinking water. There is nothing currently for irrigation waters, but there have been quite a few studies in recent years showing that these cyanotoxins can be pretty nasty, and they can hang around in the soils, and they can even be taken up by the roots, and you can even wash the plant, but you could still, if you're eating, say, that lettuce, you can still ingest that cyanotoxin even if you did wash it.
And how long, is there a time, like I know for E. coli, there's timing where, they don't want you to irrigate with water, up, like you have to not irrigate for like a month before or something. I mean, I guess that those kind of parameters exist in terms of cyanotoxin as well.
So this is still kind of like a new area.
Okay.
To my knowledge, there are no regulations for the cyanotoxins in irrigation waters.
Gotcha. Okay. Interesting. Let's see. Does the farmer need to do this pond water analysis for irrigated food crops or any type of crop?
So I guess it's a pretty similar, right?
Yeah.
So technically, like I said, from my knowledge, I don't believe there are any rules in place for pond water, for irrigation pond water, but they do have drinking water levels. And I'm not sure, I can't remember off the top of my head if there is levels for recreational water or not. I'm almost positive that there isn't, but I think it's just the drinking water at this point.
Okay. Have there been closures of, this is my own, like, I don't have the knowledge of Maryland. I know for sure that people talk about this in the West, cyanobacterial blooms, and that they close waterways and tell you not to let the dog go and swim. I mean, is this something that is cropping up in Maryland, or?
Yeah, so I'm not sure about this spring, but I know last spring, Annapolis had one of the largest dinoflagellate blooms they have ever seen. And
Wow.
Everybody was so confused because, it was coronavirus. People were kind of hunkered down at that point, and still it was a massive toxic dinoflagellate bloom right by the Bay Bridge, and you could see it by satellites. It is something that does affect the area.
Yeah, interesting. Do you think that trend is affected? I mean, of course, everything is probably affected by climate change on some level, but have we seen more incidences, I guess, with warmer waters? Is that?
I think it's probably relating more towards the increase in like runoff and nutrients coming off of farms. But there's no doubt that if temperatures would increase, there's probably some type of growth benefit there as well.
Gotcha. So we may be remediating the problem a little bit with at least in Maryland, with nutrient management planning. Maybe.
Yeah.
Yeah.
I actually learned about that in one of my classes, and there has been good progress with the nutrient management in the Bay, specifically Maryland's area.
cool. Oh, and then Emily commented, doesn't the Food Safety Modernization Act have some requirements, I guess, related to cyanotoxins?
So FSMA, I believe, is currently under, my boss, Dr. Pachepsi, would know better than this, but so the FSMA is currently under like a reassessment right now. And I believe that was more geared towards microbial counts and microbial water, like pathogens, E. coli. That is definitely in the old FSMA, but I believe they are currently working towards redoing the Food Safety Modernization Act.
Yeah.
So it's currently in the works of being refreshed.
Gotcha. Very cool. Well, thank you so much for a really nice talk and some really interesting data. So yeah, I think that's all of the questions we had. If you get a moment, please, we encourage you, please go and give Jackie some feedback via the evaluation form that Marty just posted. And otherwise, we'll plan to see you at 1 o'clock next Wednesday for the rest of seminar. So very good. Well, thanks again. Thanks for finishing us up strong.
Thank you.
Great. Good job. Thank you.